Okay, let's get started again. Okay, so just to summarise what I was saying there about cycling of nitrogen and phosphorus in the oceans. So it appears that at local scales, nitrogen disappears before phosphorus disappears. So that means that leads us to believe that nitrogen is potentially the proximate limiting nutrient of productivity in the oceans because it disappears before phosphorus does. However, there are mechanisms by which we can get new nitrogen into the ocean, and that's by nitrogen fixation. So, nitrogen gas is 80% of Earth's atmosphere, so the cyanobacteria that are able to use that nitrogen can contribute that as a new nitrogen source to the surface ocean. But really the only source of phosphorus to the oceans is by weathering and river runoff, and at this point in Earth's history we're basically in equilibrium between the cycling of phosphorus in the oceans and the input of new phosphorus by river runoff. So I talked about the redox states of nitrogen in the ocean. Nitrate is a more abundant source of nitrogen in the oceans because the oceans, because they're oxygenated, tend to drive dissolved species to their most oxidized state. So nitrate is the most abundant form of nitrogen in the oceans. However, it's energetically expensive for phytoplankton to take that nitrate and incorporate it into cell parts. So if it's present, they would much prefer to use ammonium because it's less oxidized. It's closer to the form in which they want to use it. And in fact, there have been experiments that have been done where you measure the uptake of nitrate by phytoplankton, and if you give them ammonium, they'll basically shut down the uptake of nitrate and they'll use ammonium instead. So this ammonium is in places like coastal upwelling systems, we're bringing actively moving nitrate from the deep waters up into the surface ocean. So that's what makes those areas quite productive. But in the open ocean gyres that are net downwelling, so I showed that plot of vertical velocities, there's no new nitrate being brought to the surface in those areas. So those ecosystems tend to be dominated by very small phytoplankton that are efficiently grazed by microzooplankton, so small zooplankton. And so that any biological material that's incorporated into those small phytoplankton cells is efficiently recycled back into ammonium. And so those open ocean gyres tend to be sort of spinning this nitrogen around relatively efficiently in the surface waters. But the total concentration of nutrients in those areas is quite low, so they tend to be low productivity. And then these upwelling regions like the upwelling systems like California and Peru and off Africa, they sustain relatively high productivity because of this active transport of nitrate into the surface waters. And it's these patterns of nutrient transport and nutrient recycling that set the global spatial distribution of productivity. So this is a map of net primary productivity. So it's moles of carbon that are taken up into biological material per metre squared per year. And I'll just introduce you to a couple of terms here. So primary productivity, PP, is photosynthesis. So that's the autotrophic process by which phytoplankton are using solar energy and taking up nutrients and packaging nitrogen and phosphorus and carbon into their cell parts. Net primary productivity, which is what this plot is of, is that primary productivity minus loss processes that are due to phytoplankton. So phytoplankton respiration is just another word for phytoplankton using some of the energy that they've generated in photosynthesis to do things like reproduce and maintain their cells. And then what we call net community production, or NCP, is basically this term minus another loss term, which is the heterotrophic respiration. So this is things like zooplankton consuming phytoplankton or bacterial processes degrading phytoplankton cells. So this is photosynthesis is what's being fueled by sunlight and nutrients. Net primary productivity is that minus what the energy the phytoplankton are using to maintain themselves. And then net community production is that minus the higher trophic levels that are consuming phytoplankton. Okay, so so that was just defining a few terms, and another term that we use is new production. So I was just talking about primary productivity, net primary productivity, and net community productivity. We use the term new production to quantify the amount of productivity that's stimulated by the input of nitrate into the system. So, so new production is basically a measure of nitrate uptake, whereas total primary production is, can be quantified as nitrate and ammonium uptake. So in the gyres, where recycling is efficient, a lot of the productivity is being generated or subsisting on ammonium. In coastal upwelling systems where there's active transport of nitrate to the surface, a lot of the productivity is subsisting on the input of nitrate. So if we define this term, the F ratio, which is the amount of productivity due to nitrate uptake divided by the total productivity, in coastal upwelling systems, this F ratio is quite high. So probably about a half of the productivity that occurs in those systems is being generated by the active input of nitrate. So you get an F ratio of about 0.5. Whereas in the subtropical gyres, where there's very little input of new nitrate, there's no active upwelling and there's very low rates of vertical mixing, most of the productivity is happening or subsisting on ammonium, and so the F ratio is probably about 0.1. So only 10% of the productivity is by the active input of nitrate. So we can also define a term that's the E ratio, which is the export production divided by the total primary production. So when I talk about export production here, I'm talking about the amount of that biogenic material that's being exported or sunk into the deep ocean. So this is basically the biological pump that I talked about before. So if we look at sufficiently large time and space scale, so if we were to average the new production or the F ratio over a large ocean basin, it should be about the same as the E ratio, the export ratio. All that means is that what goes up must come down. So any of the new nitrate that's brought into the surface waters gets packaged into biological material and then starts to rain out. If those terms weren't equal, if the new production was not balanced by the export production, then we would either start to deplete the nitrate concentrations in the surface waters, or we would start to accumulate nitrate in the surface waters. So they're at sufficiently large time and space scale. So if we look at a seasonal cycle or a large ocean basin, the amount of export has to be about equal to the amount of nitrate input from below. Okay, so that was a section about nutrient cycling in the ocean. So I'd like to talk for a few slides about just in a bit more detail about the organisms that are actually doing that nutrient cycling, and that's the phytoplankton. So these are photosynthetic single-celled organisms of the order of, say, anywhere from about 1 to 10 microns in size. And here's a couple of the major groups. On the left-hand side are the diatoms. So I mentioned before that they're single-celled organisms that also have a silica or a glass shell around the outside of their cells. So that means that they have an absolute requirement to take up silicon, and they're important cyclers of silicon in the ocean. In the middle panel here are what are called the dinoflagellates. So these are actually motile organisms. They have flagella that they can use for moving in the water, and they can also, many of these are not solely photosynthetic. Many of them actually consume other phytoplankton, so they also use the flagella for sort of trapping prey. These are, so if you've ever heard the term red tide, that's what these organisms are. So it's the dinoflagellates that make those red tides, because some of them are red pigmented. Over on the top right here is an example of a coccolithophorid. So this is the sort of the poster child for calcium carbonate synthesizing phytoplankton in the ocean. So they have, like the diatoms that have silica shells around the outside of their cells, these guys have calcium carbonate cells. So they're efficient cyclers of carbon, because in addition to creating cell parts out of carbon, they also have calcium carbonate around their outside of their cells. And they're very common in high latitude environments, so the Southern Ocean and the places like the Bering Sea. And in the bottom right here is an example of probably the most abundant photosynthetic organisms in the subtropical gyres. These very small cells that are about a micron or less. Cyanobacteria, Pleurococcus is an example of one, and another is Synechococcus. So these are very small cells. Because they're small, they have a relatively large surface area to volume ratio. So if you think of the surface area as the mechanism by which nutrients can get into the cell, and if you think of the volume of the cell as the demand for those nutrients, these guys, because they're small and because the way that surface area and volume scale with radius, they have a really very high surface area to volume ratio, so they're efficient at taking up very low concentrations of nutrients. And they are the main photosynthetic organisms in the subtropical gyres. And because they're small, they're efficiently grazed, and that's what I was talking about before, this recycling or this spinning of nitrogen in surface waters. Here's another example of another photosynthetic organism. This is a primnesiophyte called Phaeocystis. These are really common in polar waters as well, so you often get quite big blooms of this organism in places like the Ross Sea. And this is a colony of about 200 of these cells, and they're sort of stuck together with in a gelatinous mass. And then these are the this is an example of the nitrogen-fixing cyanobacteria that are very common in places like the subtropical gyres. Also, you often see blooms of these in places like off the northwest of Australia. So they're filamentous cyanobacteria that have the enzymes that can break down this triple bond in dinitrogen gas, and so they're the ones that can get dinitrogen out of the atmosphere and act as a source of nitrogen to surface waters. Okay, so I'm going to talk now for a few slides about the seasonal cycle of productivity in the oceans. And this is another example of a process by which we can get those two important resources, dissolved nutrients and light, in the same place for phytoplankton to use those resources and generate high productivity. So we're going to talk about the role of light and nutrients in photosynthesis. We're going to talk a little bit about the importance of zooplankton as a loss term for photosynthesis. And then the lab this afternoon is going to be running through a very simple model of the cycling of nitrogen and phytoplankton and zooplankton in the surface waters of the ocean. So the lab this afternoon is closely related to the next few slides that I'm going to talk about. Okay, so if we want to represent photosynthesis in the ocean, basically phytoplankton are taking inorganic carbon and combining it with light energy and water to make carbohydrates and evolve oxygen. Or if you look in the textbook, this is the way that they write out the photosynthetic equation. This is not strictly correct, in that probably the most abundant form of inorganic carbon that phytoplankton are using in the oceans is not dissolved CO2 gas, it's bicarbonate ions, so HCO3 with a 1- charge. So yeah, the carbon source can be dissolved CO2, carbonate or bicarbonate, but the most abundant form of carbon in the oceans is as the bicarbonate ion. So I said that the supply of carbon is never limiting. The amount of carbon in the oceans is always sufficient for photosynthesis. It's the concentration of other nutrients like nitrogen or phosphorus, or in many parts of the ocean, iron, that limits primary productivity. And as I said before, the amount of productivity on a global scale that happens in the oceans is about the same as what happens on land. So the terrestrial photosynthesis and oceanic photosynthesis contribute about the same amount each to carbon cycling or oxygen production, depending on what currency you want to think about it as. And if we were to compare, so these numbers here are the billions of tons of carbon that are being fixed out of the dissolved phase and into the particulate phase per year. The amount of CO2 emitted by the US in 2006 was about 2 petagrams or gigatons of carbon. So these are massive numbers with regards to the cycling of carbon on Earth. So the phytoplankton are green because they contain chlorophyll. The role of chlorophyll in the cells is to act as the antenna for absorbing solar radiation. And that's what phytoplankton use as the energy source for carrying out photosynthesis. And this just shows in the top plot here the absorption of different wavelengths of light by chlorophyll. So chlorophyll appears green because it doesn't absorb in the green, these mid-wavelengths here. It absorbs strongly in the blue, and it also absorbs in the red. So can anyone tell me why it might be advantageous for phytoplankton to absorb strongly in the blue? Think about propagation of light through the ocean. Yes. Yeah, blue. So blue penetrates more deeply into the ocean. All of the reds disappear in about the upper metre of the ocean, whereas blue light can penetrate much more deeply. So if you're a phytoplankton cell trying to carry out photosynthesis and you're at 10 metres depth, it's pretty useless trying to absorb just in the red because there's no red light left. That's why they absorb strongly in the blue. Phytoplankton also contain other pigments that absorb in other wavelengths. They're not strong drivers. Those pigments are not strong drivers of photosynthesis. A lot of those pigments do other things. So for instance, they can act as sunscreen, or they to sort of protect the phytoplankton from high light levels. But these other pigments don't contribute significantly to photosynthesis. So I showed at the beginning global maps of chlorophyll concentration in the surface waters. So that tells us something about the total amount of phytoplankton that are present in surface waters. It doesn't tell us, just looking at the chlorophyll concentration, doesn't really tell us how rapidly they're cycling carbon or how rapidly they're photosynthesizing. So to do that, we have to look in more detail at the transport of carbon from the dissolved phase into the particulate phase. And the way that we've been doing this for probably about 60 years now is by tracing the movement of radioactive carbon. So what we do is we can take a sample of seawater, so a couple of 100 mils or maybe a litre or whatever. I mentioned before that bicarbonate is probably the most abundant form of carbon in the ocean for photosynthesis. So what we can do is we can add some radioactive bicarbonate to a water sample, and then we can track the movement of that radioactive carbon from the dissolved phase, bicarbonate, into the particulate phase. And this is just the equation that we use to do that. So we get a seawater sample, we add some radioactive bicarbonate, we let it incubate for a couple of hours or a day, and then some of that radioactive bicarbonate is going to be taken up into cell parts. We can filter a water sample, measure how radioactive that water sample is, and that'll tell us how much of that radioactive bicarbonate has been taken up. And here's an example of how we do this at sea. So we take water samples through depth in the water column, and then we can, if the ship's not going anywhere, you can put them back on a wire into the ocean and to simulate to the light levels or the depth at which they came from. Or if you have to go somewhere, you can put that water sample in an on-deck incubator, and you can shade the surface of that incubator to simulate the light level at which that water came from. And then you can, that way you can calculate or derive the vertical profile of how fast those phytoplankton are incorporating carbon into their cell parts. So this speaks to what I was just talking about before. This shows the attenuation of light down through the ocean, and it shows that the blue wavelengths in the 400s penetrate more deeply than do the greens or the reds. So the reds are basically gone after a few metres, and that's why it's advantageous for phytoplankton to absorb strongly in the blue. So what I'm going to talk about now is the consequences of this exponential attenuation of light through the water column for the vertical profile of productivity. And so what we'll see is that in many places of the ocean, rate of productivity decreases exponentially through the water column, driven mostly by the exponential decrease of light through the water column. And we can quantify the exponential attenuation of light through the ocean by the diffuse attenuation coefficient, which is often denoted K. And here we're talking about K par, where par is photosynthetically available radiation or photosynthetically active radiation. It's basically the integral of light from 400 to 700 meters, so the entire visible spectrum. And what this table shows is that the more turbid, or the more sediment or biological material that a particular part of the ocean has, the higher the attenuation coefficient, which means that the light doesn't penetrate as far. So we can quantify the attenuation of light in the ocean by this K-par parameter, or by the depth at which we see 1% of the surface light. So in very turbid parts of the ocean, you don't have to go very deep before we've only got 1% of surface light left. So for example, in the Amazon River plume, which is has a lot of sediment being washed into the surface waters, you only have to go down half a metre before you've only got 1% of the surface light left. So that's an example of a place where there's a lot of sediment being put into the ocean, but places where there's high productivity, like the Antarctica during the spring bloom, or the certain coastal upwelling systems, the light is attenuated rapidly because of the high concentrations of phytoplankton that are in the ocean. So what this means for photosynthesis is that there's only, when there's rapid attenuation of solar radiation, there's only a small part of the upper ocean that can sustain photosynthesis. Okay, and that's all related to the attenuation of solar radiation. So if we were to look at the amount of productivity as a function of light, so here at high light levels, you can imagine we're at the surface of the ocean, decreasing down to low light levels where we're going deeper in the ocean. But at first, as we increase solar radiation, the amount of productivity increases linearly. But then the photosynthetic apparatus of the cells starts to saturate, and as you give them more light, the amount of productivity starts to level off until we get to this level called maximum productivity, or PB max. Then there are examples of types of phytoplankton and regions in the global ocean where, as the light increases even more, the phytoplankton cells start to get damaged by these higher light levels, so they can't use all of that light that's entering the cell, and that starts to have a negative impact on photosynthesis, and we call this photoinhibition. So we can quantify the amount of productivity as a function of light using these curves, and using alpha here, which is the linear slope of the first part of the photosynthetic spectrum. There's maximum value here, and also this photoinhibition parameter. We can create an equation like this which describes the shape of this curve. So what this basically shows us is how productivity varies as a function of light, and we know that light is decreasing as we go down in the water column in the ocean. And then different species will have different shapes to this curve. So certain phytoplankton species that are adapted to high light environments, so for instance the tropics, will be able to take a lot of solar radiation and use that for photosynthesis, and it'll only be at very high light levels that they're actually start to get damaged, whereas places of the global ocean where its light levels are relatively low, so you can think about perhaps the high latitudes in the winter months, those phytoplankton are adapted to low light concentrations, and you don't have to go to very high light levels before they start to get damaged by solar radiation. So then, now we're going to bear in mind that this light spectrum here, or this variability of light, happens in the vertical in the ocean, not just in different places of the ocean with the seasons. So we can use what we know about the vertical profile of light to quantify the vertical profile of photosynthesis. So now here we've got some actual values on the on the x-axis here. So 1,000 micro-Einsteins per meter squared per second would be a bright sunny day in the tropics, going down to very low light levels here. So this is one particular type of phytoplankton that has this response for photosynthetic rate as a function of light. But this is a linear scale here, and we know that light decreases exponentially in the water column in the global ocean. So what happens if we plot this on a log scale? And that's what that plot looks like now. So this would be from the surface with exponential decrease of light through the water column, and we see that the shape of this curve as a function of light now on a log scale. And now, so if this is the surface of the ocean, this is depth, we can sort of flip this plot around to show us what the vertical profile of photosynthesis might look like in the ocean, and that's what this is here. So we're going from the surface with depth, and we've got some photoinhibition at the surface, but basically we've got an exponential decrease of light and photosynthesis going down through the water column. And remember, before I was talking about net primary productivity and net community production. We can also plot on here those loss processes. So this red line here is supposed to be the consumption of photosynthetic energy by phytoplankton to maintain their cell processes. And then this blue line here is consumption of photosynthetic energy by higher trophic levels. So the green is basically the gain term, so how we're making new phytoplankton, and the red and blue lines here are how we're destroying phytoplankton or consuming phytoplankton energy, photosynthetic energy. So in the 50s, Sverdrup formalised this idea to try and understand the seasonal cycle of productivity in the oceans. So this is just another representation of that. We've got exponential decrease of photosynthesis as we go down in the water column. So this particular formalisation doesn't have any photoinhibition at the surface. And then we have this invariant loss term that doesn't change with depth because it's not dependent on light. So Sverdrup came up with the concept of the critical depth, which explains to us the trade-offs between these gain and these loss terms. And I'm going to show that in a couple of different ways. So here's this plot here is showing the depth of mixing occurring down here, and it's showing the gain terms and the loss terms for photosynthesis and respiration. So what Sverdrup said was that there's a critical depth where if you add up all of these loss terms, they're equal to the integral of the gain term. So I'll just show that in a different way here. So here's the instantaneous values for those two processes. So we've got exponential decrease of photosynthesis with depth. So that's your gain term, that's making new phytoplankton. And then we've got this invarying loss term or consumption of phytoplankton energy, photosynthetic energy, that doesn't vary with depth. And on the right-hand side, so this is the instantaneous on the left, on the right-hand side we're integrating those properties. So the photosynthesis term decreases exponentially with depth, so its integral increases asymptotically with depth. The loss term, the consumption of phytoplankton, does not vary with depth. It's constant with depth, so its integral increases linearly with depth. And then we get to this place called the critical depth, where the integral of the gain term is equal to the integral of the loss term. So if the mixed layer were here at about 80 metres, so we're essentially sealing off this upper part of the ocean, that's what the mixed layer is. At that point, in that mixed layer, the gain term is equal to the loss, the integral of the gains is equal to the integral of the losses, and so the system is imbalanced. All the new phytoplankton that's being generated by photosynthesis is being consumed by those loss processes. If the mixed layer were deeper, say at about 100 metres, we're sealing off the upper 100 metres of the ocean here, the integral of the loss term is greater than the integral of the gain term, so more phytoplankton are being consumed than are being produced. And so if we were to seal off the upper 100 metres of the upper ocean, there would be a net loss of photosynthetic material. If the mixed layer were shallower, say about 50 metres, so we seal off the upper 50 metres of the ocean, then the gain terms are greater than the loss terms, so we could have a net increase of phytoplankton biomass, and that's basically what a phytoplankton bloom is. Okay, so that just says this in a few bullet points here. The mixed layer seals off the upper part of the ocean. If we integrate photosynthesis and respiration in that mixed layer, if the mixed layer is deep, then phytoplankton are exposed to low light levels on average. They're being mixed down very deeply. And so the amount of photosynthesis that's occurring relative to the loss terms is less, so that there can't be a net gain of photosynthetic material. But as the mixed layer gets shallower, we're sealing off a shallower section of the upper ocean, and the gain terms are greater than the loss terms, and so we get net accumulation of biological material. And this is the original data that Strutton collected. They went out into the North Atlantic and stayed on station for several weeks or months, and investigated the interaction between the mixed layer depth and the accumulation of phytoplankton biomass. So in the shaded area here is the critical depth. So that's the, you can think of that as basically the depth to which light is penetrating. These vertical lines here are the mixed layer depth, and then these column pots up on the top are the accumulation of phytoplankton types, and then also the accumulation of their grazers or their consumers. So what we see is that when we get into here, it's late March, early April, as the mixed layer gets shallower and this critical depth is getting deeper, light's penetrating all the way through the mixed layer, and so we start to get accumulation of phytoplankton biomass. And after we get accumulation of phytoplankton biomass, their consumers start to increase in their population as well. So this was formalised in Strutton's theory of the critical depth, and it basically explains to us how we get high productivity in the spring and summertime in the temperate and subtropical oceans. And that's just, this is a representation of how that occurs throughout the season. So if we start out in the winter time here, the mixed layer is cold, and it's also being mixed very deeply. It's eroding down into these deep waters, which are cold. That's why the mixed layer becomes cold. But also recall that down in these deeper waters is where we have high nutrient concentrations. So high nutrients are getting mixed into the surface waters, but the phytoplankton can't take advantage of them because the average light to which they're exposed is too low. As we go into the spring and the summertime, the warmer air temperatures and less storm activity makes the surface mixed layer warmer and also shallower. So the phytoplankton are now mixed less deeply, and so they're exposed to higher light levels, and so they can take advantage of those light levels and those nutrients that have been mixed into the surface waters to generate high productivity. So then as we go into the spring and the summer, we have this shallow mixed layer, high light, beginning with high nutrients, and so we get a bloom occurring. And that's just shown in a different way here. So this is from when I used to teach in the Northern Hemisphere. It's a Northern Hemisphere example. So winter time is at the beginning of the year here. So in the winter, we have strong winds, deep mixing, it's mixing high nutrients into the mixed layer, but the phytoplankton, that's this black line here, phytoplankton aren't able to take advantage of those nutrients because they're being exposed to, on average, very low light concentrations. As the mixed layer starts to get shallower, the phytoplankton are exposed to higher light levels, and they can start to draw on that nutrient resource and generate high productivity. So we see a bloom of phytoplankton, so high levels of chlorophyll or phytoplankton cells, which starts to deplete that nutrient resource. And then, as I showed in that previous plot, the zooplankton sort of realise that there's a food source there, so they start to increase their population. So it's the grazing by zooplankton and also the depletion of nitrate that starts to diminish the bloom. Then we go into the sort of a stable situation through most of the summer, and then if there's any sort of smaller storms in the fall that generate some mixing of nutrients, we can sometimes have a small bloom in the fall, and we're going to investigate that using the model this afternoon. Okay, so to summarise these few lectures on the critical depth, we have gain and loss terms. Photosynthesis is making new phytoplankton, and consumption of phytoplankton by zooplankton and remineralisation by bacteria is the loss term. So if we integrate the gain term and the loss term over the upper ocean. If the mixed layer is very deep, then the loss term is probably going to exceed the gain term, and you won't get net accumulation of phytoplankton biomass. But if the mixed layer becomes shallow, then the gain term probably exceeds the loss term, and you can get net accumulation of phytoplankton, and that's a spring bloom. Now, that's a nice story, but there's been a lot of work lately, actually, that's been starting to challenge this idea. And in fact, the Ocean Sciences meeting that we went to a couple of years ago, there was an entire session devoted to alternate explanations for why blooms occur in the temperate ocean in the spring and the summertime. So this is, I'm not going to go into any detail on this, but this is even after 60 years or whatever, this is still an active area of research trying to understand what generates high productivity in certain parts of the global ocean in the spring and the summertime. Okay, so what I've been talking about there is basically this productive region that's happening in the surface waters, and what are the physical and chemical processes that regulate that productivity. This is, as I said before, this is often referred to as the biological pump, because at some level that primary productivity is essentially sucking CO2. If enough productivity happens to decrease the CO2 concentrations in the surface waters to a point that they're lower than the atmospheric levels, then there's going to be a net flux of CO2 from the atmosphere into the ocean that's then going to be packaged into phytoplankton and potentially exported into the deep ocean. So I've talked about the seasonal process that limits or regulates this productivity. What I wanted to talk about now is the important role of iron in limiting that productivity, and hence limiting the export of biological material into the deep ocean. So I had a question before about the role of iron fertilization as a geoengineering technique. So are there areas of the ocean that we could go and fertilize with iron, stimulate productivity, and help stimulate this biological pump to take CO2 out of the atmosphere? And and that has been an area of much interest in over the last couple of decades. So what I'm going to talk about now, and I think I'll do about 5 minutes on this to get started on the story of iron, then we'll break for morning tea. So as I said before, actually one thing that I should say is that nutrient concentrations are depleted in the surface ocean by photosynthesis, and then that biological material is exported out of the surface water. So it could be as dead phytoplankton cells, or it could be as zooplankton poop, which zooplankton have consumed these phytoplankton and then excreted them out of the surface waters. So as this biological material is falling down through the ocean, it's being remineralized by bacterial activity back into the dissolve phase. So that's what causes the low nutrient concentrations at the surface waters and the higher nutrient concentrations at depth. So especially in most of the global ocean, the average depth of the ocean is about 3,500–4,000 meters, and this material is sinking out probably at the rate of about 100 meters per day. So there's a lot of time for that bacterial activity to occur. So most of the biological material that gets packaged into phytoplankton cells gets remineralized back into the dissolve phase as that stuff is sinking out of the surface waters. It's only a relatively small amount that gets stored in the sediments for geological timescales, thousands or millions of years. If we were to try and change the efficiency of the biological pump to sequester carbon, we're really just talking about a very small fraction of that biological material that's making it into the sediments, and that's one of the impediments, I guess, for iron fertilization or any type of fertilization of the ocean as a geoengineering technique. It's only a relatively small amount of this carbon that's making it into the deep ocean and getting stored, which is where we want it to be if we're going to keep it out of the atmosphere permanently. Okay, so I'm going to talk now about the important role of iron in regulating productivity of the oceans. So there is iron is one of the micronutrients. It's present in nanomolar concentrations, or micromoles per meter cubed concentrations in the global ocean. One of the reasons that iron concentrations are very low in the present-day ocean is that iron is very insoluble in oxygenated seawater. And perhaps I'll just illustrate that with this plot here. So this is look at CO2 concentrations in the atmosphere and oxygen concentrations in the atmosphere over 3.5 billion years of Earth's history. So about 4 billion years ago, phytoplankton evolved oxygenic photosynthesis. So they evolved the capability to use sunlight to synthesize oxygen. So at that point, there was a lot of dissolved iron in the global ocean because the oceans were not oxygenated yet. So then along come phytoplankton, and they work out how to synthesize oxygen out of inorganic carbon and sunlight. And as soon as they start, so in this period here, the oceans are starting to get oxygenated. It's not until a billion years or so later that we start to see oxygen in the atmosphere. But the phytoplankton basically made the oceans oxygenated, which made the iron insoluble. So back here, they've evolved photosynthesis, and they've evolved these enzymes, which, by the way, require a lot of iron to carry out photosynthesis. And then they've basically done them themselves out of a source of iron by oxygenating the ocean. And it's in this part of Earth's history that we have things like the banded iron formation. So when the oceans became oxygenated, almost all of the iron that was dissolved in the ocean precipitated out into geological features. And so phytoplankton were left with a very low iron ocean. And so they've evolved these enzymes to perform photosynthesis that need iron. And now all of a sudden they've got very low iron concentrations. So they've had about 2 or 3 billion years to try and work out another way that doesn't require iron to carry out photosynthesis, but they haven't cracked that problem yet. Is one way you could look at it. And as they evolve photosynthesis, we also see a decrease in CO2 concentration in the atmosphere. So yeah, iron is very present at very low concentrations in the global ocean right now. Zana did the calculation that you'd need about, I think it's about 50 Olympic swimming pools of seawater. If you extracted all of the iron out of 50 Olympic swimming pools of seawater, you'd have enough to make one nail, one metal nail. Right? So it's required for processes like electron transport in photosynthesis. Actually, nitrogen fixation that I was talking about before is also a very iron-hungry process. So you often see nitrogen fixation in places where there's active input of iron into the ocean. So typical surface concentrations of iron are about half a nanomolar, and typical limiting concentrations, so when iron gets to about 0.2 nanomoles per kilogram, it starts to be limiting for phytoplankton photosynthesis. So you can see that the typical concentrations in the ocean are very close to the typical limiting concentrations for phytoplankton. So there are many areas of the global ocean where iron is low enough to start limiting photosynthesis. So iron's required at, I talked before about the ratio of uptake of nitrogen and phosphorus. A similar ratio exists for iron uptake. So when iron is present at levels below that ratio, it will be limiting to photosynthesis. And so wherever we find excess macronutrients like nitrogen or phosphorus hanging around at the surface of the ocean, that's a hint that there's something that's limiting phytoplankton uptake of those nutrients. And in many areas of the global ocean, that's the concentration of iron. It's iron that's limiting the amount of photosynthesis or nitrogen uptake that occurs. And so here's that map that I've shown several times already. In the Southern Ocean, the tropical Pacific and the North Pacific, if we go out and measure nitrogen concentrations, there's extra nitrogen lying around in the surface waters that hasn't been taken up by phytoplankton. And we now know that the reason that's occurring is because iron concentrations are really low in these parts of the ocean. Okay, so let's break there. So Melissa, we're having like coffee now, right, for a half hour? Or? Yeah. Okay. But anyway, so we'll break until 1/2 the hour, and then I'll continue talking about iron limitation. All right, so what I'm going to do now is I'm going to finish up talking about iron, the role of iron in the ocean, and I'll touch a little bit on potential geoengineering aspects of iron in the ocean. Then I'm going to talk a little bit about measuring chlorophyll from satellite because there's an aspect of the class for the QMS students that you're going to be using satellite chlorophyll data to describe variability in your favourite part of the ocean. But also just in general, measuring chlorophyll from satellites has become a really standard tool in oceanography and a really important way that we can analyse the broad-scale productivity of the oceans. And I'd like you to, even if you don't use satellite chlorophyll data in your research, I'd like you to gain an appreciation of how we do it and the limitations of that method. And then I'll close with just a couple slides on sort of introducing the lab for this afternoon. Okay, so we left off talking about the properties of iron in the ocean, and so I mentioned how phytoplankton evolved oxygenic photosynthesis that uses enzymes. So that when I say enzymes, the enzymes that move the electrons around in photosynthesis require a certain amount of iron. And if phytoplankton don't have enough iron, they won't be able to synthesize those enzymes and they won't be able to perform photosynthesis. So anywhere in the global ocean where we see extra nitrate or phosphate or silicate hanging around in the surface ocean, that's a clue that there's something limiting the productivity of the phytoplankton's ability to carry out photosynthesis. And in many areas of the ocean, that limiting property is the concentration of iron. Okay, so here's this is a clue to the iron-limited areas of the ocean. So we see in the North Pacific, to a lesser extent in the tropical Pacific, and all of the Southern Ocean, there's excess nitrate at the surface that's not being taken up by phytoplankton. And so during the 90s, especially with regards to the North Pacific, there was a lot of work that went on to try and understand what is it that's leading to this excess nutrients hanging around at the surface. I should say, or maybe it was sort of in the early 90s that this came to a head. For the longest time, it was impossible to properly measure iron in the ocean because it's present in extremely low concentrations, and all of the stuff that we go out and make measurements with in the ocean is made of iron. So, ships are made of steel. We have instruments on metal cables that we put over the side. So it wasn't until the 80s when a group at Oceanic Marine Labs in California, headed by John Martin, developed the techniques to measure iron properly in the ocean that it was then that we were able to work out exactly how low the iron concentrations in the global ocean are. And I think I said that incorrectly before. I think the calculation that Zano made was actually you'd need 500 Olympic swimming pools of seawater to be able to, if you extracted all the iron from 500 Olympic swimming pools, you'd be able to make one nail. So it's incredibly low concentrations, and for the longest time, our measurements of iron were contaminated. But this group, Oceanic Marine Labs, went out, and this is from a paper that they published in 1997. It was all of the iron, so it's also incredibly hard to make these measurements, so there aren't a lot of iron data in the global ocean. This was all the data that they had up until that point in 1997. And so, this is, I haven't actually shown any profiles yet of dissolved nutrients, but this is a typical nutrient profile in the global ocean. It's the nutrients are low at the surface because they're being taken up by biology, and as that biological material rains out of the surface ocean, it gets remineralized back into the dissolved form. So you could probably do a similar plot for nitrate, although nitrate would be about, so this is nanomolar concentration, so 10 to the minus 9 moles per kilogram, moles of iron per kilogram of seawater. If you were to do a similar plot for nitrate, you'd have probably about 2 to 5 micromolar, so about 10,000 times more concentrated at the surface, going down to about 30 micromolar at depth. So remember I said that a typical value that might start to limit phytoplankton productivity would be about 0.2 nanomoles per kilogram, and so a lot of the global ocean is around about that limiting value. So there's a lot of places in the global ocean where iron is at sufficiently low levels that it limits photosynthetic productivity. And the major sources of iron to the ocean are, one major source is river runoff. So iron is the second or third most abundant element in crustal material. So anytime you get crustal material, so dirt, dust, sediments, being put into the ocean, that can act as a source of iron to the ocean. But remember that iron is very insoluble. So of any of this dust that's making it into the the surface waters of the ocean, probably less than 1% of the iron contained in that dust can be solubilized into seawater to be biologically available for phytoplankton. But dust, so dust blowing off the deserts like the Sahara and the Gobi and Australia, one giant desert, that can act as a source of iron to the atmosphere, to the surface ocean. So notice that the Southern Ocean, the tropical Pacific, and to a lesser extent the North Pacific are all areas that don't get very much dust deposition from the dust that blows off the deserts. Okay, so these places where excess nutrients are found at the surface became known as high nutrient, low chlorophyll HNLC provinces. So all that means is that there's high nutrients at the surface, but there's low chlorophyll concentrations, which leads us to believe that something is stopping the phytoplankton, the chlorophyll, from taking up those high nutrients. And so the three major biomes in the global ocean where this occurs is the North Pacific, the tropical Pacific, and the Southern Ocean. So like I said, during the 80s, and it sort of came to a head with a couple of special issues of Limnology and Oceanography, a journal in the early 90s, there were several hypotheses that were explored to what was the process that was leading to these HNLC regions. Some people said that it was grazing, so this seems like it might be true in the subarctic Pacific that there's relatively small phytoplankton that are efficiently grazed by the next trophic level up, and so it's that grazing or that that munching of the grass sort of that's keeping the phytoplankton levels low. Others thought that it was the what I was talking about before, low average light levels. So the phytoplankton were just, especially in the North Pacific, we have pretty deep mixed layers. So it was deep mixing that was causing light limitation, that was limiting the uptake of phytoplankton. But then the group out of Moss Landing, after they made some careful measurements of iron and saw how low the concentrations really were in the global ocean, they said that the ratio of iron to other nutrients like phosphorus or nitrogen was so low that it was preventing the uptake of those other nutrients. So there was a special issue of Limnology and Oceanography, and I think it was in 1991, where all of these potential candidates for limiting productivity or generating these HNLC regions were explored. So there was papers on grazing, keeping the phytoplankton low. There was papers on light limitation, and then there was this famous paper by John Martin, it's simply called The Case for Iron. They kept arguing it. People didn't believe them, and then they basically later on they published another paper. We still say it's iron. Aren't you listening to us already? And actually, John Martin was a really interesting guy. He passed away in, I think it was 1996, but he had polio when he was a kid. So he was in a wheelchair, but he would still go to sea, and he was the driving force between this sort of behind this revolutionary group at Moss Landing Marine Labs that developed these iron clean techniques. Okay, so eventually people started to believe them. They went out, not only did they make measurements of extremely low iron concentrations that they thought were limiting to phytoplankton growth, they also took water samples and added iron to those water samples, and they saw increased chlorophyll. So they saw that if you fertilise small bottles of seawater with iron, you can stimulate phytoplankton productivity, and they'll take up those extra nutrients. So, but there was there was still some doubt about, they did these bottle experiments, but that's just a bottle. There could be something else that's being introduced into the bottle that's leading to the high productivity, or maybe when they do an experiment in a bottle, they've somehow killed or excluded the grazers. So there was mounting momentum to go and investigate iron fertilisation at very large scales, and I'll talk a little bit about that. So John Martin also, beyond just trying to explain what was causing the current day regions of HNLC ocean globally, he also looked at the paleo record, and he noted a relationship between dust and Earth's climate. So this is data from the Vostok ice core. So here we are at 0 is the present day, going back 400,000 years, and there are now ice cores that go back more than twice as long as this. So in the top panel here, we've got a proxy for global temperature. In the middle panel here, we've got a proxy for global CO2 concentrations in the atmosphere. And in the bottom panel here, we've got dust in the ice core. So these are, the CO2 concentration is just the CO2 concentration in gas bubbles in the ice. This temperature is probably from an oxygen isotope proxy. But even though Antarctica, from the map I showed before, gets very low dust input from atmospheric sources, it still gets enough that you can see some dust in these ice cores. So what John Martin, and actually by the way here, to have a look, go back 400,000 years, and CO2 has never got above about 280. And if you go back, you can go back more than a million years, and it hasn't exceeded 280. I'm sure someone knows what we're up to today. Yeah, so up to about 400. So we're all the way up here. So go back a million years in Earth's record, and we haven't seen CO2 concentrations like we have today. Anyway, John Martin noted that at times of Earth's history where it was very dusty, there was also low CO2 in the atmosphere, and the Earth was cooler. And in places in Earth's history where there wasn't much dust, CO2 was higher and the Earth was warmer. So he was suggesting that this dust was acting as a source of iron. It was fertilizing the ocean. Phytoplankton were taking up those extra nutrients. They were also sucking up CO2 out of the atmosphere, leading to a cooler, lower CO2 planet. So this is just a schematic of what how he envisaged this. So in glacial periods of Earth's history, it's cold, but it's also drier because a lot of Earth's water is locked up in ice caps and glaciers, so the deserts are actually bigger. So when the deserts are bigger, that's potentially a bigger source of dust to the ocean. So more deserts, more dust going into the atmosphere, more dust going into the ocean, alleviating iron limitation, stimulating primary productivity, sucking up CO2, some of which goes into the deep ocean and some of which gets stored in sediments. So I had a question at the break about how much of this organic material that's produced from photosynthesis makes it into the sediments in the deep ocean. And I don't know if Zana wants to correct me here, but I think it's probably, especially in the open ocean where it's 3,000 or 4,000 meters deep, it's probably about 1% or less of this biologically fixed carbon makes it into the sediments. So this was John Martin's iron hypothesis, how dust in the atmosphere could be regulating climate. Notice that this is a positive feedback loop. There's nothing in this diagram that shows how we get out of this loop. But John Martin suggested that it was the delivery of iron to the oceans that was at least partially responsible for these fluctuations in CO2 between glacial periods, the cold periods, and interglacial periods, the warm periods. There's been subsequent work that's shown that biological productivity in the ocean can probably only account for about half of this variability. So it's not, John Martin's iron hypothesis is not accounting for all of that 100 ppm change between glacial and interglacial periods. So John Martin is, he's famous or perhaps infamous for saying, give me half a tanker of iron and I'll give you an ice age. So he, on the back of an envelope essentially, he calculated how much iron you would need to go into the Southern Ocean, fertilize the phytoplankton and get them to suck up all of those extra nutrients and get them to suck up 80 ppm of CO2. So he said this more or less facetiously at a Journal Club lecture at Woods Hole. He calculated that with 300,000 tonnes of iron, Southern Ocean phytoplankton would bloom and remove 2 billion tonnes of CO2. So, do you know this movie, Dr. Strangelove? So he, doing his mad scientist act, he said that with half a tanker of iron, he could give us another ice age, after which they went and had a beer outside the Redfield lab. So he wrote this up in an article. It's quite interesting. So here's me having a beer on the lawns outside the Redfield lab 21 years later. Okay, so after that work, finally John Martin convinced the grazers and the light people that it was potentially iron that was limiting productivity in these parts of the ocean. So starting in 1993, the first open ocean iron fertilization experiment happened. So they went out, so mostly a group of US scientists went out to the tropical Pacific, one of the iron limited regions of the global ocean, and they fertilized a part of the ocean that was about 10 kilometres by 10 kilometres with iron. And they saw, briefly, they saw an increase in chlorophyll, and they saw a drawdown of nutrients and a drawdown of CO2, but something happened in the parcel of water that they'd fertilised got subducted under another parcel of water, and so it was too dark down there for the bloom to continue. But then in 1995, they went and did a follow-up experiment, again in the tropical Pacific, and that was much more, I guess you could say, successful. I mean, they stimulated, again, a bloom about 10 kilometres by 10 kilometres, and confirmed iron limitation, not just in bottles, but in the real ocean. Since then, there have been iron fertilisation experiments in the other iron-limited areas of the global ocean, the North Pacific and the Southern Ocean. And Xana and I were on a voyage in 2002 that went down and fertilised 2 patches in the Southern Ocean, and I'll just talk briefly about that. So this is me and Xana here. So we went out to 2 different places in the Southern Ocean. One where there was high nitrate, or both patches that we fertilised had high nitrate, but one had high silicate and the other one had low silicate. And what we were trying to do there was to investigate the role of diatoms, because it was thought that if we fertilised an area that was high in nitrate but low in silicate, we might not get a bloom of diatoms, whereas if we fertilised a patch that was high in both, we would probably get a bloom of diatoms. And we wanted to see what the consequences might be for export of biological material out of the surface waters. Remember before I was saying that diatoms have a glass shell around their cells, and some people think that that makes them better ballast for exporting biological carbon out of the surface waters, just because they're heavier. Okay, so we went out to the Southern Ocean. We had these about 5,000 gallon tanks that we filled up with a couple hundred kilos of iron sulfate, a couple hundred litres of hydrochloric acid, and a couple thousand gallons of seawater. We put the hydrochloric acid in there to maintain the pH low, so that that helps the iron be bioavailable. And then we pumped it off the back of the ship. This is a plastic fish that we attached to basically a fire hose going off the back of the ship. And even though this thing's plastic, it started rusting because of all the iron that was sort of flooding on top of it. We used this to try and keep the hose at 10 metres depth while we went and fertilised. Now, I was just having a very interesting discussion with Alex about the role of iron in land plants. And if you were to investigate the role of iron in stimulating productivity of land plants, it's a little bit easier than doing it in the ocean, because you can fertilise your cereal crop or whatever, and you can go back a week later and the plant's probably still going to be in the same place. Whereas in the ocean, everything's moving, so we need some way to keep track of this fertilised patch. So what we do is we chuck a drifter in the middle of our patch that we fertilise, and hopefully that drifter will move with the patch of ocean. Another way that we keep track of where we fertilise is we put an inert tracer in as well. So there's this chemical sulfur hexafluoride, which is biologically inactive, but you can measure it at extremely low concentrations. So when we're putting out the dissolved iron, we also put out a very small amount of this sulfur hexafluoride. So we can measure the concentration of sulfur hexafluoride to know that we're still going over the top of the patch that we've fertilised. So after the phytoplankton take up the iron, we wouldn't be able to tell where the patch was just by the concentration of iron. We use the concentration of sulfur hexafluoride. So this is one of the patches that we fertilised, and it was about 15 kilometres by about 15 kilometres. And after we fertilised the patch, we fertilised it sort of in this fashion. We're sort of mowing the lawn back and forth, pumping the iron out the back of the boat. Then we go back and we make some measurements to try and see what the iron has done to the phytoplankton. So we measure sulfur hexafluoride, and we see high levels of sulfur hexafluoride, which means we're going over the correct patch. If we measure the CO2 concentration of the surface waters, we see lower CO2 concentrations in the fertilised patch, because the phytoplankton have had their photosynthesis stimulated, and they're starting to suck up CO2 out of the surface waters. This is just another way of looking at that drifter that I just showed you, as well as marking the centre of the patch. So we had real-time communication with this drifter so that we knew where we were. It also has instruments on it, including an instrument that measures carbon dioxide. So this is at the start of the fertilisation, that the CO2 in the ocean was about the same as the CO2 in the atmosphere, and after a couple of days we'd drawn down about 10 parts per million of CO2 in the middle of the patch. So the phytoplankton are having their productivity stimulated, and they're sucking up CO2 out of the surface waters. And because the CO2 is now lower in the ocean than it is in the atmosphere, CO2 can potentially go into the ocean, and that's sort of one of the things that we're trying discover here. Also, this plot here just shows the iron concentration. So the mixed layer was about 50 metres deep, and you see in the fertilised patch we've got higher iron concentrations compared to the control station that we did outside. And then this plot just shows the changes in phytoplankton biomass through time. Each one of these profiles here is going forward a couple of days, and basically we see in the mixed layer, the upper 50 metres, we see increased chlorophyll concentrations as a result of the iron fertilisation. So the iron fertilisation has stimulated the phytoplankton. I could have also showed a similar plot here for nitrate concentration. They're also drawing down nitrate, and they're sucking up CO2. Okay, so to summarise the role of iron in the global ocean, the concentration of iron is extremely low in most of the global ocean, and there are at least 3 very large oceanic provinces where the concentration is potentially limiting to phytoplankton photosynthesis. It's about 0.2 nanomoles per kilogram. This is in the North Pacific, the tropical Pacific, and the Southern Ocean. They're called HNLC regions for high nutrient or high nitrate, low chlorophyll. And around about 1990, John Martin suggested that iron delivery to the ocean as dust was regulating climate, and that's the Vostok ice core data that I showed. So since 93, there's been at least a dozen of these open ocean iron experiments that have confirmed the role of iron in regulating productivity in these 3 large provinces. But I should say that John Martin's quip about, give me half a tanker of iron and I'll give you an ice age, hasn't proven to be true. The efficiency so we have seen when we fertilise with iron, we have seen productivity stimulated, and we've seen some uptake of CO2. But if you try and measure the amount of particulate carbon that's being exported out of the surface waters, it's actually very small compared to the numbers that John Martin used. So we need more than half a tanker of iron to give us another ice age. And so I had some questions in the breaks about the potential of using this as a geoengineering technique, and for a while it was talked about as a geoengineering technique to try and suck CO2 out of the atmosphere and mitigate global warming. But this factor that the amount of export that we've seen is relatively low has meant that it seems to be not feasible to use this as a geoengineering technique. But it's still mentioned in terms of things like selling carbon credits. And for instance, there was a somewhat controversial experiment a couple of years ago where a community in British Columbia paid a entrepreneur from California I think it was about $2.5 million to go and fertilise the ocean off their coast to try and stimulate salmon fisheries, and they were also trying to sell carbon credits. So I think you understand what I'm talking about there. That, if you suck up CO2 in one part of the ocean, you can sell that right to pollute basically to someone else who's emitting CO2. Now there's also other. Yeah, sorry. I was just about to say, yeah. So there's potentially several negative impacts. So probably the most important being that everything in the oceans is connected, right? So, and I mentioned at the start of the very first lecture that for instance, in the Southern Ocean, that nitrate that's not taken up gets subducted into mode waters that then spread throughout the rest of the global ocean, and modelling studies have suggested that unused nitrate or unused nutrients that go into the mode waters and then go into the rest of the ocean potentially stimulate or are responsible for about 70% of global primary productivity. So if you fertilise with iron in the Southern Ocean and you start to suck up those extra nutrients, then those mode waters are going to be subducted with fewer or lower concentrations of nutrients, and that's going to impact total global primary productivity. There's also other potential negative impacts. Wherever you get high productivity in the global ocean, when that biological material rains out, it's consumed by bacteria. I've mentioned that several times. That consumes oxygen. So below productive parts of the global ocean, like, for instance, the Peruvian upwelling system, you get extremely low oxygen concentration in mid-waters. And so if you were to stimulate productivity in places like the Southern Ocean, you're probably going to generate large low oxygen areas, which has significance for other marine life. Some people think that there are certain phytoplankton that make toxins, and like I was talking about red tides before. So phytoplankton like that, if they're stimulated by iron fertilisation, then that could lead to a toxic bloom, which has implications for the food chain. I've done some work that has shown the impact of these blooms on ocean physics. So mostly we think about ocean physics in regulating productivity. So I've been talking a lot about how physical processes either mix nutrients to the surface or upwell nutrients to the surface. So a lot of times we think about physics as the driver of biological productivity, but I've done some work that shows if you stimulate blooms, it actually impacts the physics of the ocean. And the way that it does that is by absorbing solar radiation. So photosynthesis is basically absorption of solar radiation. If you put more chlorophyll in the surface ocean, you're going to absorb more solar radiation, so you're going to warm up the surface of the ocean, where otherwise that solar radiation would have penetrated into the deep ocean. So with a colleague, I did some modelling that showed if you put a massive bloom in the North Pacific, for instance, you'll warm up the surface ocean by about 1 degree. You'll also warm up the atmosphere over that ocean by about 1 degree Celsius. And then you also change the cloudiness and the humidity, and so it has a significant impact on ocean physics. So those are all potential negative impacts of geoengineering, and we need to understand those processes properly if we are going to use this as a geoengineering technique.